Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for your upcoming examination. So I have compiled so many important instrumentation questions which can be asked in your any of the exam like CSIR, and if it is merged with CSIR DBT both one exam, so then it can be asked. It can be asked in GATE exam and different PSC based exams. So the first question is, the absorbance A is related to transmittance by which of the following formula? Out of all the four formula, the exact formula for the relation between absorbance and transmittance be. So absorbance is the logarithmic inverse of transmittance. It means if transmittance is 100%, absorbance is 0. You can say that understand this by taking this example. Suppose if you are having a solution whose concentration you want to measure. So if you have measured the concentration in the spectrophotometer, then if you have observed that the transmittance is 80%, only 80%, so it means that 20% had been absorbed and rest of the is 80%. So whatever is absorbed, it has to be subtracted from 100, so you can get the transmittance. So if transmittance is 100, it means nothing has been absorbed. So if transmittance is 100, it means the absorbance is nothing, 0. So this is the relationship between absorbance and transmittance. Absorbance refers to the amount of light absorbed by the sample. It means 20% of the light has been absorbed by the concentration of your sample or the particles present in your sample, and then 80% is the transmittance. And if the absorption is 0, then you will expect 100% transmittance. So this is the right answer. If you are asked that transmittance is itna percent, then tell me the absorbance. So you only need to subtract it. Suppose if transmittance is 80%, then what will be the absorbance? 20. If it is 60%, then what will be the absorbance? 40%. So you only need to subtract from 100. Now the next question, chromatography. So in HPLC, which of the following is used as a stationary phase in reverse phase chromatography? So firstly, you should know about the reverse phase and the normal phase chromatography. So the reverse phase chromatography, that is RPC, which is a obviously liquid chromatography technique that separate molecule based on their hydrophobic interaction with a stationary phase. So in RPC, the stationary phase is non-polar and the mobile phase is polar. So you can remember this. In reverse phase chromatography, the stationary phase is non-polar and the mobile phase is polar. It means hydrophobic molecules in the mobile phase will bind to the non-polar stationary phase, while the more polar molecule elute more quickly because your mobile phase is polar. So the right answer is C18 bonded silica. In reverse phase chromatography, the stationary phase is non-polar, that is C18, and the mobile phase is polar. So you have to remember that this is the reverse of the normal phase chromatography. So in this, stationary phase is non-polar and mobile phase is polar. Now moving on to the next question. Electrophoresis. SDS in SDS page provides which of the following? So what is the role of SDS in SDS page? So DNA denaturation, positive charge to proteins, uniform negative charge, protein folding. So in SDS page, SDS is used. SDS is a detergent. So SDS is a sodium dodecyl sulfate and it plays a crucial role in denaturating protein and separating them based on their molecular weight. So what SDS do? SDS is a detergent that disrupts the non-covalent bond with protein. So SDS is responsible for breaking down the non-covalent bonds within the protein and unfolding them into a linear chains, and also it coats the protein. Suppose this is the protein, it provide negative charge to the protein, so it can migrate towards the positive electrode during electrophoresis, and this is how the separation takes place. So SDS in SDS page provides uniform negative charge. SDS denature protein and coats stem with negative charge, so separation is by size only. So this is the right answer. can also come to the answer by option elimination method. DNA SDS is related to protein separation. Positive charge to protein, no. SDS provides negative charge. Uniform negative charge is right in protein folding, no. It does not support in protein folding. It denatures protein. So this is the right answer. Rate in ultracentrifugation depends on. So the sedimentation rate is governed by the size of your sample, shape and density, as well as the viscosity of the sample. So it depends on B, the viscosity of the medium. So sedimentation rate in ultracentrifugation refers to a speed at which the particle in a solution move and settle under the influence of higher centrifugal forces. So सेडिमेंटेशन मीन्स जब भी हम सोल्यूशन में हमारे कुछ पार्टिकल्स होते हैं उनको अगर हम लोग स्ट्रॉन्ग सेंट्रीफ्यूगल फोर्स देते हैं तो पार्टिकल्स क्या होते हैं, दे एक्सपीरियंस अ फोर्स एंड अलाउड दिस फोर्स पुश देम अवे फ्रॉम द सेंटर ऑफ रोटेशन. यानी इस वजह से क्या होता है, जो पार्टिकल्स हैं वो सेडिमेंट होने लगते हैं, सेटल होने लगते हैं बॉटम में ऑन दी viscosity of the medium, size of the particle, shape, density of the particle. So the right answer is viscosity of the medium. Now moving on to the next question. Microscopy. Which microscopy is best for viewing live unstained cell? So you need to mention that which microscopy is involved in this, in which you don't need to stain the cell. So a phase contrast microscope is a specialized type of microscope which is known to do this. It can observe the transparent, unstained specimen like living cell, and this by having a visible difference in the brightness without needing to stain the sample. So this photography is particularly useful in biological research for observing the cellular structure and the dynamic processes in live cell. We don't need to stain the cell. So the right answer is phase contrast microscopy. Now next question, PCR related. What is the role of Taq polymerase in PCR? So the role of Taq polymerase is to, so Taq polymerase is what? It is a thermostable DNA polymerase that is crucial for PCR. It is derived from the bacteria Thermus aquaticus. So this is a thermostable DNA polymerase which is very much needed for PCR. Without that, PCR cannot be done, as it is capable of withstanding the high temperature needed to denature the DNA during the process. First step is DNA denaturation. So we do it at a very high temperature, and for that we need the Taq polymerase. So it is related to DNA synthesis at higher temperature. So the right answer is option C. Now next question. In ELISA full form is enzyme linked immunosorbent assay. In sandwich ELISA, the antigen is captured between two antibodies labeled directly attached to the plate, detected using substrate. So you need to study about the ELISA because it is very important. So many questions are asked in CSI related to the ELISA and different techniques. So the sandwich ELISA is a highly sensitive technique for detecting the antigen. It detects the antigen and it utilizes the two antibodies to sandwich the antigen between them, like this way. If this is an antibody, they use to sandwich the antigen in between two antibodies. So it is captured between the two antibodies. This is the right answer. So the correct answer for this question is option A. Now moving on to the next question. So next question is biosensors related. So in glucose biosensor, the enzyme used is glucose oxidase. We have heard this many a times. So glucose oxidase react with glucose to produce hydrogen peroxide, which is detected electrochemically. So in glucose biosensors, you should know about how glucose biosensors work, because this is asked so many times. So in glucose biosensor, they use the biological component like the enzyme glucose oxidase. Glucose oxidase is used to reacts with the glucose in a sample, and then the reaction produces a measurable signal in the form of hydrogen peroxide. Like it can generate signal through different electric current generation, and that is why it will be proportional to the exact glucose concentration in your blood. So these are all important. So when glucose is present, this GO, glucose oxidase, will do what? It will facilitate a reaction where glucose is oxidized to gluconic acid and oxygen is reduced to hydrogen peroxide. And then this is detected, and this is the basis of the detection in glucose biosensors. So right answer is glucose oxidase. Now moving on to the next question. Spectrophotometry. A double beam spectrophotometer uses two beams of light. So why it uses two beams of light? To compensate for solvent and instrument drift. So the reference beam passes through a blank, helping correct fluctuation in the light source and baseline drift. So in double beam spectrophotometer, we use two beams of light. So what is double beam spectrophotometer? So in double beam spectrophotometer, which is a type of spectrophotometer that uses two light beams to measure the absorbance, or you can say transmittance, of a sample. So it uses two light beams to offer enhanced stability and accuracy compared to the single beam model spectrophotometer. So one beam passes through the sample and the other act as a reference, allowing for real-time compensation for the fluctuations in light source intensity and other factors. So in double beam spectrophotometer, one light is passed through the sample, यानी एक बीम ऑफ लाइट सैंपल से पास होता है एंड एक बीम ऑफ लाइट आपका रेफरेंस जो होता है उससे पास होता है, सो दैट इट विल अलाउ फॉर द रियल टाइम कॉम्पनसेशन फॉर फ्लक्चुएशंस इन लाइट सोर्स इंटेंसिटी. यानी इससे पता चलेगा, जो इससे हर पॉसिबल एरर को एलिमिनेट करने की कोशिश की जाती है, यानी जो भी लाइट फ्लक्चुएशंस होने के चांसेस हैं, हमारा जो भी लाइट सोर्स है एंड उसके अलावा जो अदर फैक्टर्स हैं, इंस्ट्रूमेंटल ड्रिफ्ट, वो कम कर सके हम, इसीलिए वी यूज टू डू दी डबल बीम स्पेक्ट्रोफोटोमीटर. सो अगर आपका एक में सैंपल है एंड एक आपका रेफरेंस है, तो यू विल गेट एन एक्यूरेट पॉइंट इफ यू आर हैविंग द डबल बीम स्पेक्ट्रोफोटोमीटर. नाउ नेक्स्ट. In ion exchange chromatography, proteins are separated based on molecular weight, no. Net surface charge at a given pH, yes. No shape, no hydrophobicity. So ion exchange chromatography is dependent on the net surface charge at a given pH. So protein binds to oppositely charged resin depending on the isoelectric point and the buffer pH. So this is an important type of chromatography which is based on the isoelectric point of the protein to be separated. So this is all for today. I hope you liked it. If you want me to make a questions series of a particular topic, you can comment below, and don't forget to subscribe the channel. Thank you so much for watching.